Take a look at this picture, and before you proceed with the video, pause and try to think about what this could be. Hopefully you thought about this. This is actually a microscope. It's one of the first microscopes created by Anton van Leeuwenhoek. Basically, it was a way to examine the cellular world. So things that were too small to see with the naked eye, with just your eyes alone, you could gather a sample, put it over this little thing called a lens. The lens would bend light a certain way and then allow you to see a larger image of what was there. So you could actually see cells with this little microscope that you could hold in the palm of your hands. So in this lesson, you're going to learn about what a microscope is, how it looks very different from the microscope we just saw in the previous slide. Microscopes look like this today, and they look even more different when you get to university. There's different types of microscopes. This is just one of the many types that exist, and this is the common laboratory type that we're going to see in high school. By the end of this lesson, you'll be able to label and explain what all the different parts of the microscope do. You'll be able to actually use a microscope to find a specimen at different powers. You'll be able to do some microscope calculations, and you'll be able to prepare your own specimen slide. So a microscope is a tool that biologists use in order to see things that are too small to see with just your eyes, with the naked eye we see. So cells happen to be one of those things that we can view under the microscope. We are using what's called a compound light microscope. The compound part means that we're using more than one lens. So there's the lens that you look into over here, and the lens that's near the specimen you're observing. So that's why there's two lenses that you're looking through at any given time. So that's why it's called compound. And it's called light because your specimen is illuminated with light. There's a light source over here that shoots some light onto the cell you're trying to observe so you can see it more efficiently. So we have a compound light microscope. There's many different types of microscopes. Some microscopes actually use electrons to view specimen. And there's a really cool one at McMaster you can see one day. But we're going to be using light. The thing you're trying to see, that's called the specimen. So whatever you're trying to observe under here, that's called the specimen. And you usually put the specimen on a slide. So this is a microscope slide. So you get a sample of cells, spread them onto the slide, and then you can put the slide onto this little part over here called the stage, and essentially you can move some knobs, adjust some knobs, and make the stage go up and down to get a nice, clear, focused image of your specimen. So before we jump into the microscope itself, we're going to take a look at some pictures of cells seen under the microscope. So when you look under the microscope, you'll basically see this circle here, and that's your field of view. That's basically the area in which you can view specimen. And here you can see a bunch of cells. This is a cell here, this is a cell here. All these little bricks are essentially cells. So take a minute to try to think about what type of cell this might be. So I'm just selecting this cell here, but they're all the same type of cells here. Is it plant or animal? And then try to label the parts. Try to name the parts that are labeled there. So after pausing the video, here are the correct answers. So this is a plant cell. The way you can tell it's a plant cell is by its shape in this situation here. Shape is not always the best way to do it, but this is a good guess to first start off. So it's a plant cell. It's a rectangular cell, and they're all very closely packed together. Now, knowing it's a plant cell, you can start to label some of the parts. So if you look at this here, this is an organelle that only plants have. And if you said chloroplast, that's correct. That's a chloroplast there. Here we're pointing at essentially nothing. So the big gooey jelliness in there, it could be cytoplasm. Some students sometimes say that if you cut the cell open, maybe you've cut the cell wall away, you're looking at the cell membrane, but you're probably looking at the cytoplasm there. And then over here, this arrow is pointing at the outside of the cell. So if this was an animal cell, you would have said cell membrane. But because it's a plant cell, you know the outside has an extra layer covering, and that's likely the cell wall. So this is an image of a cell taken under the microscope. You're not going to see color like that all the time unless you stain it. Stain it is when you put a chemical on it so you can add some color to it and get better contrast in the image. And you won't see chloroplasts like this under every microscope. You really need to have powerful microscopes with good resolution and good magnification. But for now, we're just getting a taste of what some pictures could look like under the microscope. We can try this one out here. So this one you'll notice also has rectangular boxes. And it's a bit of a trickier one, but if you guessed Plant cell, you're correct. You can try to label the different parts. So you can pause the video and try it on your own. But you'll notice there's a darkly stained area here. And that darkly stained area is probably the nucleus. So when you see a darkly stained area and there's only one of them, that's usually the nucleus. Over here and over here you have arrows pointing at spots that are very similar. This is the outside spot, that's the inner part there. So the outside spot is probably the cell wall, and the inner part there is probably the cell membrane. And over here, this is a bit tricky. You'll see it says vacuole. It's very hard to see, but there's a central vacuole here. I would probably label that as cytoplasm, because you can barely see the vacuole in this image here. So if you said cytoplasm, that would have been correct as well. You can try this one on your own. You can see there's actually two cells here. If you guess this is an animal cell, that's correct. It doesn't have that rectangular box, so it's probably an animal cell. Either way, it's not a plant cell. There's a darkly stained spot here, so you're probably looking at the nucleus. Here you're pointing at the outside. There's no cell wall for an animal cell, so probably a cell membrane. And here you're pointing basically at the ooey gooey jello-y stuff. That's probably the cytoplasm. And there you have it. So that's just some practice labeling cells that you might see under the microscope. It's a bit tricky at first because you're used to seeing cartoon images, but eventually when you look at the cartoon image and you look at the actual cell under the microscope, the shapes and the placements start to make more sense, and you can more easily make that connection. And of course, the most important slide of all, a lovely lame joke as we proceed into the microscope. Why did the germ cross the microscope? To get to the other slide, microscope slide. We can pause for laughter. So what we're going to see in this first part of the video is the microscope itself, the compound light microscope. We're going to take a look at the different parts of the microscope and see what they do. You can follow along on the handout that's provided to you, or that's in the learning goal area or course calendar. You can take notes about what all the different parts do. What's important is that after this video, you're able to label a picture of the microscope, and you're able to tell me what all the different parts are responsible for. You should also be able to do the same thing if you have an actual microscope in hand. So make sure you get practice with that as well. So these are all the different parts here. We'll start off down here. This is called the base. This basically supports the entire microscope. On the base, you can see a little switch here. The switch might be in different spots sometimes depending on a microscope you have. This switch will control the light source. So when you turn this on, you of course have to plug it in, turn it on. Light comes out of here and goes into here to illuminate or shine on the specimen here. You also have what's called a diaphragm. The diaphragm, you can adjust how much light goes through, so it's not too bright or it's bright enough for light to hit the specimen. So light source, diaphragm here. Where you put the specimen, this black piece here is called the stage. Okay, because the specimen is the star of the show, that's called the stage. Remember, you're never putting cells directly on the stage. What you're doing is you're putting the cells on a slide. So this is a slide over here. And then you would add a drop of water or chemical to make better contrast when you're trying to view it. And then you'd put what's called a cover slip on top. So you have your slide, specimen, cover slip, and you would put that on the stage. To make sure the slide stays in place, you would put the slide underneath these stage clips. And these are the stage clips here. And you'd aim to try to get your specimen in this middle area here because that's where the light comes out of. Now, your eye would come here. That's where you're viewing from. So that's called the ocular or the eyepiece lens. And then that connects to the rest of the microscope using the tube. So the tube connects the eyepiece lens to the rest of the microscope, specifically to the other lenses that you're going to view from. And these are called the objective lenses. So the fact that we have two lenses means that we magnify twice. We make the image look bigger 2 times. The eyepiece lens in the microscopes we use have a 10 times magnification. So if something is 1 centimeter long, it'll look 10 centimeters long through this eyepiece microscope. Then you choose one of the 3 objective lenses here. There's a low power, a medium power, and a high power. And some microscopes will have more. but it's usually written on it 4, 10, or 40. And so what that means is that one of these would magnify by either 4, 10, or 40. So if I choose the low power, that magnifies by 4 times, the medium power by 10 times, and the high power by 40 times. But think about this carefully. Suppose I'm looking through the eyepiece and through the low power. I'm magnifying 10 times 4, 40 times. If I'm looking through the eyepiece and the medium power, I'm magnifying 10 times 10, 100 times. And if I'm looking through the eyepiece and high power, I'm magnifying 10 times 40, 400 times. So you have basically magnification happening twice at the eyepiece lens and at the objective lens. To switch between different objective lenses, the low, medium, and high, you use what's called a revolving nose piece. So this basically just spins around so you can click the lens that you want to view the specimen to into place. And you got to hear it click into place there. And then we have the arm. The arm is basically responsible. It's a spot for you to hold the microscope from. So usually you hold the microscope from the arm, and then you have a hand at the bottom of the base there to support the microscope as well. So those are all the different parts of the compound light microscope. You have to make sure that you know how to label a microscope like this, but also how to explain what all the different parts do. There are 2 more parts that I didn't explain yet, and I'll go over that right now. We have the adjustment knobs. There's the coarse adjustment knob, and then there's the fine adjustment knob. So the coarse adjustment knob is used to move the stage up and down. So if your stage is down here, you can turn the coarse adjustment knob to bring it up closer to the lens. And this moves the stage a little bit as well, but not much. It's called the fine adjustment knob. So the idea is that when you first try to find your specimen under the microscope, you use the coarse adjustment knob to bring it as close to the lens as possible. And then it'll be blurry at first when you find it. So you'll turn these fine adjustment knob to focus the specimen more so it's not blurry. An important thing to understand is that the coarse adjustment knob, you only turn it when you're on low power. Because if you turn it on other powers, medium or high, you can bring the stage too far up and it'll hit the medium or high lenses, and that can cause them to crack and break. So we only ever change the coarse adjustment knob, turn that on low power. You can turn fine adjustment on all powers because it doesn't really move the stage that much. But coarse adjustment knob only on low power, so make a note about that. So what we're going to do is we're going to take a look at how to hold a microscope properly, and also how to use a microscope properly. That'll be in the next video. So in this video we saw how to label images of cells taken by a microscope. It's called a micrograph. We saw the parts of the microscope and what they do. And then in the next video we're going to take a look at how to hold a microscope properly and also how to use a microscope in order to find a specimen.